To protect wildlife, conservationists need to first answer what might seem like a simple question. Which species actually live in the habitats they're trying to save? This question might be difficult to answer, especially in dense and complex habitats like rainforests or shifting landscapes around cities. Ecologists have a long list of methods at their disposal: radio collars, camera traps, acoustic monitoring, and even good old-fashioned direct observation.
Okay, I see where it went down.
But the newest tool has the potential to be the most comprehensive yet. By collecting small samples of soil, water, or air, scientists can now create a genetic roster of every species that calls an environment home. It's a tool known as environmental DNA. Here in Detroit, environmental DNA, or eDNA, is revolutionizing our ability to survey wildlife. Dr. Naima Harris and Dr. Jane Hallam are tracking how predators, like coyotes, travel through the urban landscape and how their range overlaps with the other wildlife, with local pets, and with humans. Camera traps have been the go-to method for this type of research for decades. But now with eDNA, scientists can detect species without ever catching them on camera. Animals shed DNA constantly, through scat, or hair, or skin cells. Samples from a single habitat might contain this so-called trace DNA from dozens or hundreds of wild species.
We get that species list from the cameras because we see the images. When we're collecting the eDNA, we also then get a species list. So that means when we've collected the soil and we've done the molecular analysis, the DNA that we've detected has come from 24 different mammal species in the city of Detroit. And some of those species we don't actually see on the camera.
eDNA isn't new. It's been used to detect wildlife since the early 2000s, but the technology continues to advance. Today, scientists can identify species using much smaller fragments of DNA than ever before. Trace DNA collected in the field is replicated through a technique called a polymerase chain reaction, or PCR. It makes millions of identical copies of the genetic material, which can then be cross-checked against a DNA database of known species.
This is a bit like forensics. Environmental DNA tends to be quite broken up because it's been exposed to, the elements, and it's been broken down by ultraviolet light from the sun. So that DNA isn't in great shape. It tends to be in quite small fragments, but that's enough for us to detect the species.
The size of those DNA fragments is key to why environmental DNA is such a powerful technique. For context, the genome of an animal like a coyote includes roughly 2.5 billion base pairs, but you don't need a complete genome to identify a sample source. Certain small DNA sequences are shared among all members of a species. They act like the barcodes you see at the store, and when fragments of eDNA from the wild are cross-checked against a database of existing barcodes, we can match the fragments to the species they came from. Early eDNA methods required a barcode 100 base pairs or longer in order to identify a species. But today, with new sequencing technology and better computing power, fragments as small as 30 base pairs are often enough for a successful species match. This has broadened the use of eDNA to some amazing extremes. Even ancient, degraded DNA from soil 2 million years old can reveal extinct species, like woolly mammoths. And in dense environments like the rainforests of Ecuador, where vegetation makes wildlife surveys difficult, eDNA has become an essential tool. Traditional genetic analysis can sample just one species at a time. Now, eDNA can sample hundreds at once. To the teams on the ground, eDNA is a low-cost, low-disturbance tool that detects species that are otherwise extremely difficult to find. From the rainforests of Ecuador to the streets of Detroit, results from eDNA are directly impacting environmental policy. Conservationists everywhere are embracing eDNA as the latest powerful tool at their disposal. Finally, a way to reveal an entire ecosystem worth of life in a single scoop of dirt. Explore these stories and many more from Wild Hope by visiting wildhope.tv or watch the full collection now right here on PBS Nature on YouTube.